introduction. And I would like to thank for the organizer to invite me to their wonderful place and to attending the wonderful conference. I enjoy it very much. So today my topic is about the biomarker study on psychiatric disorders. It's a really big topic, so I'm mainly focused on the genetic analysis and regarding the transdiagnostic dimensional phenotypes across 3 common mental disorders, which include schizophrenia, bipolar disorder, and major depression. Okay, as we all know, when we talk about the genetic analysis, the phenotype we used previously mainly focused on the diagnostic categories, which means the patient with or without these mental disorders. And according to the diagnosis criteria such as DSM or ICD. But there is always an issue for phenotype. As we celebrate the human genome success, and also the researchers has been pointed out, the most important is the correction of the human phenotypes. We should, according to the agreed upon parameters and standards. So it has been mentioned this for about more than 10 years now. And for my group, we have been focused on those 3 common mental disorders, as I said, including schizophrenia, bipolar disorder, and major depression. And our research outlines many by correcting the patient with those disorders and also their relatives and their health controls. We do the clinical assessment, and also we have been doing the imaging assessment in all those patient relatives and controls, and also including the cognitive assessment. And we also collect their biological samples for DNA and RNA extraction and collect their plasma for some factor analysis. And the purpose for those data collection is try to establish a multi-level, multimodal phenotypes, because my training in UK is mainly focused on genetics analysis. I would like to see if any phenotype people might call the intermediate phenotype or endophenotype. I would like to call the multimodal phenotype if they can save for the better phenotype for my genetic analysis. So previously, my group has been doing quite a lot of analysis focused on one disorder or another, schizophrenia, bipolar, or MDD, and for the cognitive function as comparison, imaging comparison, or some genetic analysis. So I don't have time to talk about the previous work. What I'm going to talk today is why I choose the transdiagnostic dimensional phenotype at the moment for my current work. So, as we know, there is overlapping clinical phenotypes across a lot of psychiatric disorders, especially for schizophrenia, bipolar. Some people would call them psychosis, and also for major depressions. If some of the patient with psychotic symptoms, they might join the group of the psychosis. And also for those patients, they have very much the clinical heterogeneity. As we can see in clinic, the patient with same diagnosis, they might have different clinical symptoms. And also for some of the patients with different diagnoses, often they share some clinical symptoms. So also for genetic study, the well-known PGC, psychiatric genomic consultant work, they have been found those 3 disorders, also including other psychiatric disorders, that may have genetically correlated. And this is a well-known publication, has been published last year in Science. This study analysis about 1 million participants, including 10 psychiatric disorders and 15 neurologic disorders. And the main finding for this study is a mega-analysis. They found there was a widespread sharing across psychiatric disorders. And also they found the MDD, the major depression, has shown the greatest genetic correlation with anxiety. And the bipolar disorder showed the greatest genetics with schizophrenia, not surprisingly called psychosis. And also the MDD was significantly correlated with the bipolar disorders as well. As we can see, all those 10 psychiatric disorders have very much correlated with each other. And also for other study, has shown that multidimensional cognitive impairment are prevalent for 3 disorders, for those schizophrenia, bipolar disorder, and major depressions, for all, with the spectrum features in a number of cognitive domains. And also, previous study also showed the shared genetic etiology between cognitive functions and mental disorders. As we can see, all those disorders have shared genetic factors with cognitive functions. So this is my student Pei-Yan Li, and she has done some work by comparing the multi-factors in plasma in patients with schizophrenia or bipolar disorder or MDD. Basically, we collected 4 groups of individuals, including 3 groups of patients and also 1 group health controls. We did a factor measurement in plasma. In total, there are 22 factors, including the immunology, circadian stress factors, and the endocrine factors, and also the neuropeptide factors. I will show you the summary result from our preliminary analysis. We can see from those 3 patient groups, they all showed the neuropeptide and also circadian factors have significantly decreased compared to the health controls. And also for those 2 patient group, including the MDD and the bipolar disorders, they showed increased immunology factors and also the endocrine factors, but those not happened in schizophrenia patient group. And we also have doing quite a lot of MRI study in those 3 patient group. The patient with those common mental disorders have significant structure and functional neuroimaging change, and compared to the controls, it is very consistent with other studies. But those different are not sufficient or not consistent to distinguish each other. And my student Min Li has done some work by comparing the bipolar depression with the unipolar depression, and she found the group difference in degree centrality of the resting state functional MRI in bipolar depression and the unipolar depression. They have some shared abnormality, and also they have some unique change in each groups, so which suggested probably only from the imaging features we cannot distinguish or just say those are the same group or different group. So for my current study, we have been focused on the cross disorders, including those 3 disorders. And the main purpose is to refine the transdiagnostic dimensional phenotypes by across 3 disorders, and by integrating the information from the clinical symptoms, cognitive functions, and the neuroimages. And furthermore, we would like to identify the genetic factors by genes or by pathways and the neural mechanisms that might give rise to the core behavioral dimensions of psychopathology. This is the research pipeline we have been doing at the moment. We continue to collect the health controls, and particularly for the patient with those 3 disorders. The patient with schizophrenia, they first absorbed drug naive, so try to involve the medication confounding effect and also the chronicity, because the schizophrenia patient they have a very long history and the chronicity, and also the medication may affect the change. And we collected the clinical cognitive MRI and also the ERP data and for deep phenotyping. And by using the deep phenotyping to do the genetic analysis, and also we use a public database like the PGC and other public database to validate our finding from our own samples. So I would like to report some very preliminary result analysis by my student. Yajin Meng has been doing quite a lot of imaging study, and Hong Yan and Yamin doing the genetic analysis mainly. And we collected about 1,053 individuals, including the schizophrenia patient, bipolar patient, and depression patient, and also the health controls. And for the assessment, as I had mentioned, we use a clinical assessment for the symptom and diagnosis. And also we use the concept for cognitive assessment. And for the imaging study, we collect the structural and functional data, including the structure VBM and DTI and the rest state functional. And this is what we have been used for the clinical assessment. And also for the cognitive assessment, we mainly use the concept and also the ways for the intelligence scale. And as for the MRI. And for the genetic analysis, we did the whole genome genotyping, the GWAS, use the Zhonghua A, B-chip, and also later on we use the global screen area. And also we did the whole exon sequencing in the same set of samples. And the QC, escape it. And what we have been doing, what we have done now, we just use the genotype we got from the GWAS, try to predict the transcription. And this is the in-sequence expression quantification of the targeted genes in the targeted brain regions. Try to see if any genes we have found by using the GWAS could be targeted in a particular brain regions with a particular transcriptome. And we have two strategies. One is from the right side, is the top-down, by using the summary statistics from the public database, such as the PGC, for those three major disorders, and to follow the common factors from the GWAS and get the expression profile. And another strategy is from the bottom up, the left side, and we use our own GWAS data to discover the transcription profile, and then to identify the genes and the particular brain regions have the particular transcription genes. And some of the results from the top-down approaches. And after, this is the method we have been used. So we try to detect the common factors underlying these three disorders. And by using the structure equation model, we found one factor model could significantly explain the latent structure underpinning those three disorders. As we can see, especially between the schizophrenia and the bipolar disorders, they're highly correlated with each other, and also with significant correlation with the major depression as well. And in terms of the genomic position, expression profile, and the genetic correlation with other traits, we try to identify the genomic variations associated with the common factors, as I showed in the last slides, and try to interpret the social loci. And this is the main result. We found there is some loci on chromosome 6p, and which spans about 3 megabase, was associated with the common factors I just showed with the genomic significance. And also the QQ plot also indicated a genome genomic inflation factors. According to the LD score regression, the inflation is mainly due to the polygenesis rather than other confounders. And also this SNPs, when we run in these regions, one of the SNPs, the 429, it is look at the intronic region of the scan12 gene, shows the strongest signal of the association. And when we look at the public database from the PGC or the UK Biobank, we found the highly genetically correlated with the multiple psychiatric, cognitive, and behavioral traits, including schizophrenia, depression, and bipolar disorders. And also we did the heritability enrichment analysis in chromosome state and the cell types under the selection pressure. We found 4 cell types as shown in the right side. And we also did the fine mapping of the social loci. With this loci, we use the biocell approach. We found the EQTL for the NF391 in multiple brain regions and also the global cerebral expression. And according to the output from the GTEx consortium website, this locus is significantly associated with the expression level of the NF391 in multiple tissue, including brain regions. And especially we found in 12 brain regions that 8 brain regions showed an increased expression of the NF391, including the amygdala and cortex, especially for the cerebrum, has showed the most significant. And then we use a transcriptome profile from those genes we have identified, try to see if we can generate the new biotypes for the patient, include the 3 category disorders. And we generate the 6 new biotypes for the patient based on their transcriptome profiles. And the left side, we found that the 6 biotypes, the top 3 are mainly schizophrenia, and the bottom 3, which including 3 disorders, just mixed. We also validate by K-means is another method, and it's confirmed our result. And now, at the moment, we are trying to annotate the newly defined biotypes in terms of the clinical features and cognitive functions and imaging. This work is still ongoing. Unsurprisingly, we found in the top 3 biotypes, there are much more severe psychotic symptoms because there are many schizophrenia patients. But for the bottom 3 biotypes, the psychotic symptoms is much smaller compared to the top 3 one. And also we are doing more analysis regarding the cognitive function and imaging and other biological features. So because of time, I think I will skip the bottom-up strategies. I will just show you the result. We found from the GWA study, one markers on chromosome 6 has a significant effect for the general cognitive functions. And also this effect has been mediated by the cerebrum. So it's really converging from our top-down strategies and also the bottom-up strategies has been showing the cerebrum we should pay more attention in psychiatric disorders. And I don't think I have further conclusion for our study, but there was widespread clinical cognitive imaging and genetic similarity, and also the difference across psychiatric disorders. It is much needed to refine diagnostic dimensional phenotypes across mental disorders in order to identify biomarkers. And I would like to thank the founding body from Chinese government and also my colleagues and students in West China Hospital, Sichuan University. Particularly, I would thank Professor Andy Gresham and Ximin Li in University of Alberta and Professor Park Xiang and his team in Hong Kong University. And thanks very much.